Good morning, everyone. My name is Weston Park. I'm a junior specialist in Dr. Gukowski's lab, and it is my pleasure to introduce Dr. Gurevich as today's distinguished speaker. Dr. Gurevich is a professor of pharmacology and of ophthalmology and visual sciences at Vanderbilt University, where he is also a Cornelius Vanderbilt Endowed Chair. He earned both his bachelor's and master's in biochemistry from Moscow State University, followed by a PhD in bioorganic chemistry from the Shemaikin Institute in Russia. He then completed his postdoc training in molecular pharmacology at Thomas Jefferson University. Dr. Gurevich's research has been foundational in advancing our understanding of arrestin proteins and their role in G protein receptor signaling. His work has revealed the molecular mechanisms of arrestin activation by active G protein coupled receptors and receptor attached phosphates. Dr. Gurevich and his team has determined structures of all 4 vertebrate arrestin subtypes, as well as the arrestin-rhodopsin complex. His lab has also pioneered the development of engineered arrestins that bind receptors without phosphorylation, revealing the in vivo role of arrestin self-association and demonstrating that enhanced visual arrestin can compensate for defective rhodopsin phosphorylation in mice, which has significant implications for both fundamental research and therapeutic strategies, especially in visual disorders. Dr. Gurevich's contributions to structural and molecular pharmacology cannot be overstated, with over 280 published scientific papers, tens of thousands of citations, and the mentorship of new students. His work continues to bridge fundamental biochemical mechanisms with translational approaches to disease. Please join me in welcoming Dr. Gurevich.
Well, thank you. I hope I won't disappoint you after this introduction. So I'm going to talk about structures, but I'm not going to talk only about arrestins. And one important thing from this slide, my short name is Seva. Don't try to say my full name. Only Russian and Polish people can do that.
Vsevolod.
Most of them.
Vsevolod, we noticed that.
Okay. So before going into arrestins, I want to talk about GPCRs or like 7 transmembrane. Come on, baby. I mean, it should. Why does it move? Okay, now it does. 7 transmembrane proteins in general. So this is the 3D structure of bacteriorhodopsin. Bacteriorhodopsin is light-driven proton pump in bacteria. And this was solved by electron microscopy at 7 angstrom resolution. And one other thing I want you to, I'm sure the structure looks pretty familiar now, but one thing I want you to notice is the year. It's 1975. Now, this is the primary structure of bacteriorhodopsin. And again, look at I don't expect you to remember the structure, but look at the year. It's 1979. So it's one of the very few proteins where the 3D structure was established before primary structure. Now, this is the organization of bacteriorhodopsin in the membrane. So what I want to point out is a pretty typical distribution of aromatic amino acids. And the lysine, where the retinal attaches, is at the 7th transmembrane helix. Now, this is the structure of bovine visual rhodopsin. And again, just look at the year, 1984. So it was established a lot later than the structure of bacteriorhodopsin. And this is the arrangement of rhodopsin in the membrane. Again, look at the aromatic residues. They are very similar to bacrhodopsin. And again, the lysine where the retinal is attached, it's at the 7th TM. So this is the structure of bovine rhodopsin, and Chris should recognize it. It came from his lab. This is actually the first structure of a GPCR resolved. And as you can see, those 7 transmembrane helices. And if you compare the structures of bacteriorhodopsin and mammalian rhodopsin, They are not identical, but you can see quite a few similarities, including the fact that retinal is attached to the seventh transmembrane helix. Now, there is a debate in the field whether these proteins emerged by convergent evolution or came from the same ancestor. Now, if they emerged by convergent evolution, then apparently like this order of helices and the fact that retinal is attached to the seventh should be important. So this was addressed experimentally. So these helices in bacterial rhodopsin were swapped every which way. This is the, in the left top corner is the wild type sequence. And you can make the primary structure, linear structure, with these helices assembled in various ways. And you can even introduce the extra helix, marked as W here. And it turns out that all these proteins are, or many of them, are functional. Some actually, as the second one here, is even better than the wild type, suggesting that the order of helices in the linear structure is basically irrelevant. The protein folds anyway. So the same group asked whether it's important that retinal is attached to the seventh helix. So they engineered proteins where the retinal was attached to different helices, and those turned out to be functional as well. So all these data suggest that the similarities between rhodopsin and bacterial rhodopsin probably are explained by a common ancestor rather than by functional convergence. Of course, we cannot prove something that happened hundreds of millions of years ago beyond reasonable doubt, but these data suggest one answer, although if you look at the primary sequence homology, no existing software can find homology between bacterial rhodopsin and bovine rhodopsin. Now, this is about rhodopsins and where all these 7TM proteins came from. And now the major breakthrough in the field where we actually got the idea that there is a family of G protein coupled receptors came when beta-2 adrenergic receptor was sequenced. It turned out that it also has the, on the right, there is hydrophobicity profile of rhodopsin and beta-2 receptor. They are similar. The receptor has 7 transmembrane helices. And in this case, the software can determine the homology between these two proteins. So this was important conceptually because it implied that all these regulatory mechanisms that were previously studied with rhodopsin apply to other G protein coupled receptors. And now we know that this is true. Now, I'm sure you saw this tree. And this is 2003, so we have a lot more structures now than labeled here. But basically, this tells you that humans have about 800 different G protein coupled receptor subtypes. Among these, about 300 are non-odorant, and pretty much all mammals have the same 300 non-odorant GPCRs. Now, most mammals have a lot more odorant receptors. We being primates, are microsmatic, so we don't have a very well-developed sense of smell. So we have only about 500 of odorant GPCRs, whereas other animals have a lot more. The record holder, I believe, is an elephant that has almost 2,000 different odorant GPCRs. Now, let's talk a little bit about GPCR signaling. So we have GPCRs that respond to all sorts of things: light, extracellular calcium, odorants, small molecules, peptides, and proteins. In all cases, active receptor activates heterotrimeric G protein, and subunits of heterotrimeric G protein, and I'll talk about it later, go on to activate various effectors. And these effectors are either enzymes or ion channels, so you have even more amplification at this stage. So speaking of amplification, so during its lifetime, activated GPCR activates anywhere from 5 to 80, obviously 80 applies to rhodopsin, G protein molecules. Activation means the exchange of bound GDP in heterotrimer for a GTP. Upon GTP binding, G alpha subunit of the G protein and G beta gamma dissociate from each other and from the receptor and go and regulate various effectors, like I said, ion channels or enzymes. So amplification of this second step is about 100 to 10,000, depending on the system. So this huge amplification, if you multiply the first step by the second step, actually gives you very high sensitivity to GPCR stimulation. In case of rhodopsin, it has been measured, and it turned out that our rods, after we fully darkened up by sitting in the dark for like 30, 40 minutes, can detect single photons. And they can detect single photons with the probability of 70, 80%, which is about the same as the best physical detectors we can make. Now, the signaling, like everything the cell does, takes energy. And naturally, the cells try to save energy. So as soon as the signal goes through, the cells have mechanisms to stop that signaling because it becomes useless. And these mechanisms, in case of most GPCRs, are the same. So this is the schematic of homologous desensitization. Active receptors are phosphorylated by specialized kinases, G protein coupled receptor kinases, GRKs for short. And then phosphorylated active receptors are recognized by arrestin, which binds to them, shields the cytoplasmic surface, thereby precluding G protein activation. So that stops the signal at this step. Now, this mechanism was established over many years. So first, rhodopsin phosphorylation was actually described first in 1972 for both frog and bovine rhodopsin. Phosphorylation of beta-2 adrenergic receptor was discovered more than 10 years later in 1983. Arrestin was also first discovered, like everything, in visual system in 78. And then it was shown to preferentially bind phosphorylated rhodopsin. And a few years later, it was shown to shut down rhodopsin signaling to transducin when it binds. And the first visual arrestin was discovered a lot later, in 1990. It was actually cloned by homology with visual. Now, what do we have in terms of desensitization? What do we have? We have 7 GRKs, which are termed GRK 1 through 7, 4 arrestin proteins. Again, systematic names are 1 through 4, but arrestin 2 and 3 are also called beta arrestin 1 and beta arrestin 2. Now, in terms of GRKs, we can feel superior to mice and rats because we have 7, and they don't have cone-specific GRK7. So they have only 6. As far as arrestins go, all animals have the same 4. Now, we have arrestin 1, which is also called visual rod arrestin, which I don't think is correct, and GRK1 in rod photoreceptors. In cone photoreceptors that mediate color vision, we have arrestin 1 and 4, 4 is cone specific, as well as GRKs 1 and 7. And in some species, like mice and rats, you only have GRK1, and in some others, you only have GRK7. And we have 2 non-visual subtypes, arrestins 2 and 3, expressed pretty much in every cell. And we also have 4 ubiquitous GRKs, 2, 3, 5, and 6. And in some cells, you also have GRK4. So every cell in our body has a number of different GPCR subtypes, 5 to 25. Your guess is as good as mine. At least 2 non-visual arrestins and 3 or more, sometimes up to 5, different GRKs. So GPCRs are very important for drug discovery and medicine in general. So their activation mechanisms were studied in detail for many years. And it turned out that activation of GPCRs structurally means the movement of transmembrane helices, which opens a cavity between helices on the cytoplasmic side. As pretty much everything, it was first discovered in rhodopsin in the visual system and later found to apply to all ligand-controlled GPCRs. So what happens upon activation, you open this cavity. So there is a place for the proteins that preferentially bind active receptors to dock. Now, I told you that GRKs phosphorylate active receptors. So how do they know that the receptor is active? And again, this piece of data on the left was done by Chris, so he should recognize it. And that was the first indication that GPCR docking actually activates GRK. So in rhodopsin, all the phosphorylation sites are localized on the C-terminus. You can cleave off that C-terminus by proteolysis, and you get a protein that can still be activated by light, but cannot be phosphorylated anymore. So if you mix this protein in the dark with GRK, and throw in the peptide that GRK can phosphorylate, pretty much nothing happens. Now, if you activate this rhodopsin by light, GRK starts phosphorylating this peptide like crazy, suggesting that docking to the active receptor activates GRK, and then it can phosphorylate whatever is nearby. In real life, nearby is usually the elements of the same receptor GRK docked to, but you can substitute the substrate artificially in vitro. Now, very recently, in 21, we finally found out how this happens. So the salt structure of GRK1 complex with rhodopsin showed that first, GRK, like G protein and arrestins that I'll show later, docks to the same interhelical cavity of the active GPCR, and that the two lobes of the kinase domain of GRK1, which are not perfectly aligned in the basal state, upon rhodopsin binding become perfectly aligned and therefore yield high kinase activity. I also, let's talk a little bit more about GRKs. So we have 7 GRKs which fall into 3 subfamilies. So the visual ones, GRK1 and 4, have very short C-termini, and those C-termini are prenulated. They are farnesylated in GRK1 and granulated in 7. This hydrophobic modification actually anchors GRK to the membrane where its substrate resides. Now, GRKs 2 and 3 have the longest C-termini in the family. They have a separate so-called Blackstream homology domain. This domain mediates GRK interaction with G beta gamma, and G beta gamma serves, which is also prenulated, serves as a membrane anchor for these GRKs. Now, GRKs of the third subfamily, GRK4, 5, and 6, have sort of intermediate sized C-termini, and they use a different way of targeting membrane. They have either amphipathic helices or palmitoylated cysteines, which help it to bind the membrane where GPCRs are. Now, one of the reasons I'm showing this DRK, comparative GRK structure is like most proteins are multi-domains. And in most multi-domain proteins, domains are one after another, like beads on a string. Now, GRKs in that sense are unique. In GRKs, the kinase domain, shown in yellow here, is actually inserted in one of the loops of the RGS domain. And RGS is a regulator of G proteins that facilitates GTPase activity of heterodimeric G proteins, speeding up their self-inactivation. So, and this structure of GRKs is conserved throughout all mammals, suggesting that this insertion occurred pretty early in evolution and was kept because it worked. Now, I also told you that arrestins preferentially bind active phosphorylated receptors. So the question is, how do they know receptor is active and phosphorylated? Now, this was addressed again with visual arrestin and rhodopsin using a direct binding assay. So obviously, phosphorylated active rhodopsin binds arrestin best. That's the red bar here. But if your assay is sensitive enough, see it's femtomolar, you can detect certain level of binding to just phosphorylated dark rhodopsin or activated unphosphorylated rhodopsin. So this suggested that arrestin can first bind rhodopsin through phosphates or through the active state. And then if both of these things are there, which can only happen with phosphorylated active rhodopsin, arrestin undergoes a significant conformational change, which brings additional elements in contact with rhodopsin. And that explains why the binding to active phosphorylated rhodopsin is a lot greater than the sum of the binding to inactive phosphorylated or active unphosphorylated. Now, this is arrestin structure. Again, Chris should recognize this. This is the highest resolution arrestin structure there is, and it came from his lab. So, and I'm showing this structure because the structure of arrestin 1 monomer was proven to be correct by many experiments using different methods. So arrestin is an elongated molecule consisting of two cup-like domains. And the C-terminus comes back, and here it is. Maybe you can see it. And resides in the cavity of the end domain. So arrestins in the basal state have two autoinhibitory interactions. One is, and I'll show the details later. So one is the 5 charged residues. Negative charges are shown in red, positive in blue, in the middle between the two domains. And the other is so-called 3-element interaction, where mediated by hydrophobic residues in all elements between beta strand 1, C-terminus, and alpha helix 1. So both of these interactions need to be destroyed for arrestin to bind a GPCR. Now, this is the structure. So I told you that this structure appears to be pretty correct, even though, I mean, despite the title of my talk. Now, this is the structure of the arrestin receptor complex. Again, it's arrestin 1 and rhodopsin. And you can see that the finger loop in the central crest goes into that same cavity where G proteins and GRKs dock. And the polar core and 3-element interaction are disrupted, and there is a domain movement, which I will show in the next slide. And I want to point your attention that here in this structure, rhodopsin engages the central crest of the arrestin molecule and the end domain where the rhodopsin C-terminus resides, and does not even touch the C domain. Can that be true? I mean, can this domain be an extra? Well, biochemical data suggests that it is not. Now, I'll start with the early experiments. People made arrestin-derived peptides and tested them for the ability to compete with arrestin and transducin that both bind active rhodopsin. Well, this first peptide, 51 to 70 on top, represents the finger loop, which we know from the structure binds, and obviously it competes with both arrestin and transducin. But the peptide at the bottom, 231 to 250, actually comes from the rhodopsin C domain. And as you can see, it also competes, suggesting that it binds and the C domain binds. Now, this is a totally different method used to ask the same question where rhodopsin binds. So this was done with EPR, electronic paramagnetic resonance. You can put an EPR probe on the chemically modified cysteine that you put anywhere in arrestin. And if it's on the protein surface, obviously it's pretty mobile. And you can tell the mobility of the probe by its spectrum. Now, in basal arrestin, it's pretty mobile everywhere. But if you bind it to rhodopsin, the mobility would change in the positions that come into contact with rhodopsin because your EPR probe would be trapped in the protein-protein interaction interface. So in blue, in the top structure is shown where the mobility actually goes down. And as you can see, it goes down in the central crest and in the end domain, which the structure predicts, but it also goes down in some positions in the C domain, which contradicts the structure. Interestingly, if you go from dark rhodopsin, shown on top, to light-activated phosphorylated rhodopsin, the mobility of the labels at the tip of the finger loop changes further, which suggests that finger loop is what Arrestin uses to discriminate between active and inactive rhodopsin conformation. And finally, before I go to the last piece of evidence, the C domain is involved in receptor binding, I need to show you this. So the top is the comparison of Arrestin 1 and one of the non-visual subtypes, Arrestin 2, and the bottom is the comparison of the structure of Arrestin 2 and 3, two non-visuals. And as you can see, they are all very, very similar. Actually, the fourth subtype, cone-specific Arrestin 4, also has essentially the same 3D fold. In fact, when we submitted the structure of Arrestin 4 to the journal, one of the reviewers said, Arrestin looks like Arrestin, so what else is new? So the similarity of the 3D fold suggests that you can swap homologous pieces between subtypes and it would not screw up the folding. So that's what was done. So Arrestin 1 loves rhodopsin and doesn't like particularly muscarinic M2 receptor. Arrestin 2 binds rhodopsin but doesn't like it as much, but it loves M2 muscarinic receptor. Now, if you swap two pieces, one in the N domain, one in the C domain, shown in green in the structure below, if you put these pieces from Arrestin 2 into Arrestin 1, you make a protein that doesn't like rhodopsin but loves muscarinic receptor. If you put the same two pieces from visual Arrestin into Arrestin 2, now it loves rhodopsin but doesn't like muscarinic receptor. So it suggests that these two pieces are responsible for receptor preference. Now, if you only swap the N domain piece, you do not get this effect. And what is shown in CPK models in the structure below are the residues critical for receptor preference, which were identified later. And again, you can see that many of them are in the C domain and just a few in the N domain, again suggesting that C domain is involved in the interaction. Now, the contradiction between biochemical, biophysical data and structure needs explanation. So here, I don't expect you to remember all these structures. I'm showing this list of all the Arrestin GPCR structures solved just to show you that there are many of them. And in all of them, the C domain does not touch the receptor. Now, what would be the reason for that? Now, let's start with the good news. So from the receptor arrestin structures, we learned what happens with arrestin molecule upon receptor binding. So one thing that happens is the finger loop, shown in red here, goes up and, like I showed, docks in the cavity between GPCR helices. The neighboring middle loop, shown in blue here, in the basal state, it overhangs the C domain, and in receptor-bound state, it moves in the direction of the N domain. One of the legs that attach the alpha helix to the body of the N domain actually changes the conformation more or less the same way on receptor binding in all arrestin subtypes. And the two arrestin domains twist relative to each other, but by anywhere from 18 to 20 degrees relative to the basal state upon receptor binding. And finally, the interdomain hinge, shown in yellow here, has various conformations in different arrestin subtypes in the basal state, but in receptor-bound state, it always assumes exactly the same conformation. Now, still, we need to explain the controversy between biochemical, biophysical data and structures. So let's look at the arrestins and let's look at these self-inhibitory activation. So in the middle between the two domains, there is what's called the polar core. So 5 charged side chains, 2 arginines and 3 aspartates that interact with each other. And the 3-element interaction with hydrophobic residues from the alpha helix, C terminus, and the beta strand 1 in the end domain is shown down below. Now, you can destabilize these interactions by reversing the charge like aspartate to arginine or arginine to glutamate in the polar core, or you can delete the C terminus and destabilize 3-element interaction, or you can, instead of deleting it, you can replace these bulky hydrophobic residues with alanines and again destabilize this interaction. So it turns out that in rhodopsin receptor, arrestin receptor structures, in all arrestins used for structural work, one or both of these self-inhibitory interactions were destabilized. And in red, I show which in each structure. Now, is it important? Does it change the function? Well, it turns out that it does. Now, in the visual arrestin, it loves light-activated phosphorylated rhodopsin. It doesn't like very much other functional states. But if you introduce a triple alanine substitution in the C terminus, it still binds best to active phosphorodopsin, but now you gain a lot of binding to phosphorylated inactive or active unphosphorylated rhodopsin. Now it's the same story with arrestin 2, which was tested with beta-2 adrenergic receptor, one of its cognate receptors. The wild type arrestin 2 also binds best to active phosphoreceptor, but it also demonstrates significant binding to inactive phosphorylated receptor, but not to the other states. Now, if you introduce triple alanine substitution, reverse the charge in the polar core, just delete the C terminus, you get more or less the same phenotype. You gain a lot of binding to inactive phosphorylated and especially active unphosphorylated receptor. So the arrestins used for structural work were not exactly functionally equivalent of the wild type forms. Now, the receptors, the same applies to receptors. So here I am showing what was changed in the receptors used in the arrestin receptor complex structures. And well, obviously a lot of things were changed, but one thing I want to point out, in pretty much all structures, the native C terminus of the receptor was replaced with multiphosphorylated C terminus of V2 vasopressin receptor. And again, how important is that, you might ask. So I'll show you the most glaring example. Muscarinic, that's number 10 here, muscarinic M2 receptor. Now this receptor, unlike rhodopsin or beta-2 adrenergic receptor, has a huge short cytoplasmic loop and very short C terminus. And all phosphorylation sites important for arrestin binding were shown to be localized on the short loop and none of the C terminus. Now in the structure, this loop was deleted and this C terminus was replaced with multiphosphorylated V2 C terminus. So the receptor that is in the structure has very little in common with the real M2 muscarinic receptor. So that probably the changes in the arrestin and receptor probably explain why the structures do not quite show what happens with wild-type proteins. Now, that's at all call, so it should be tested, and it was again tested with arrestin 1 and rhodopsin. And I won't bore you with all the examples, I'll show the two most clear ones. So this serine and glutamine in the structure are pretty far from rhodopsin, which is shown in green here. Yet if you mutate these residues, you significantly affect the binding of wild-type arrestin to phosphorylated and unphosphorylated light-activated rhodopsin. Now, this is the opposite example. This isoleucine and leucine are pretty close to hydrophobic residues in rhodopsin in the structure. Again, green, rhodopsin is green and arrestin is yellow here. But if you replace these bulky hydrophobics with an alanine, essentially eliminating the side chain, nothing happens to the binding of wild-type arrestin to rhodopsin. Now, these are examples, but this is the list of the whole thing. Various parts of arrestin were mutagenized, and the effects of point mutations were tested on the background of wild-type arrestin and pre-activated arrestins with C-tail deleted or triple alanine substitution, which are functionally equivalent to the arrestins that are used for structure determination. It turns out that in more than 2/3 of these cases, the effect of the mutations on the background of wild-type and enhanced arrestin are different. So overall, that suggests that the structure of the mutant arrestin bound to mutant rhodopsin and other arrestin mutants bound to mutant receptors does not recapitulate the structure of the wild-type arrestin bound to wild-type receptors. Now, this statement implies that the complex of arrestin with receptor can exist in different flavors or shapes. Is that true? Well, it appears to be. Now, in both structural studies of arrestin-rhodopsin complex, distance measurements were made between certain points in rhodopsin and certain points in arrestin. Now, this was made by double electron-electron resonance, DER for short, and this method measures the distance between two spin labels in a protein or a complex. And now you probably know that distances can be measured by resonance energy transfer, fluorescence or bioluminescence, breadth of FRET. And here I'm showing you measurement by DER. So breadth and FRET, if you have multiple distances, what they give you is the average. And this average is about as informative as an average fever level in the hospital. The beauty of DER is if you have multiple distances, you see them all. And if you look at the DER distance profiles obtained by DER, you can see that in all cases, in all measurements, there are multiple distances between rhodopsin and arrestin. Now, the most probable distance always matched the crystal, to the delight of crystallographers, but the other distances show that the complex exists in different shapes as well that coexist with that crystallized shape. Now, the same question was asked with parathyroid receptor and arrestin 2. So this was done in cell, in vivo, in a real situation where these proteins work. So in the receptor on the cytoplasmic side, cysteines were introduced in different places. And in arrestin 2, a reactive, cysteine reactive unnatural amino acid was introduced again in different places. And these things were combined in all variations. And when these two residues come close together, you get a crosslink. If they don't, you don't. So using this method, 136 proximity points between receptor and arrestin 2 were determined. Now, molecular dynamic simulations show that all of them cannot possibly be explained by any individual shape of the complex. But if you consider all possible shapes, then they explain all the proximity points. Again, suggesting that the complex of arrestin 2, this parathyroid hormone receptor, also exists in many different shapes in the cell. Now, finally, let's look at structures. So structures can be determined by two methods, X-ray crystallography and cryo-EM, which is becoming prevalent method nowadays. Now, in the crystal, the complexes of different shapes cannot possibly co-crystallize. The crystal selects a particular shape. Otherwise, you get very low resolution. Whereas in cryo-EM, you work with individual complexes. So if you analyze hundreds of thousands of these particles, you can actually resolve variations of the complex. So this was done with CPR1 receptor and Arrestin 2, and it was done very recently in 25. And the authors resolved 4 different shapes of the complex. As you can see on top, they're pretty similar, but they vary in arrestin orientation relative to the receptor. So again, this method tells you that the complex of the same arrestin with the same receptor can have different shapes. Now, what does the direct binding assay, the data of which I showed you, actually detect? It is based on gel filtration, on separation of arrestin bound to rhodopsin containing membranes, which are big, from free arrestin, which is small. So basically for this assay, it doesn't matter how exactly arrestin is bound, what shape the complex has, as long as it elutes with membranes, it is counted as bound. So basically it gives you the sum total of all possible shapes of the complex that survive this gel filtration. Now you can, structures are fairly fashionable now, and here I am sort of bad-mouthing them, so why? I need to have an explanation for that. And so I'll show you the example where structures were experimentally proven to be wrong. 3 different labs, 1 in Germany, 2 in the US, crystallized visual arrestin 1. And all of them got more or less the same shape of the arrestin tetramer, shown here on the left. Now, using DIR, interprotomer distances were measured in many positions, and then the modeling says that the solution tetramer matching these distances has a very different organization, shown on the right here. Now you can say that, well, modeling is not data, so you have to prove it. And it was proved experimentally. In the model, phenylalanine 197 and phenylalanine 85 from different protomers come close together and apparently mediate the interaction. So if that is true, then modifying these residues would change the self-association of arrestin 1. So in the central graph, in the red, wild-type arrestin 1 is shown, and you can see that it is readily self-associates. If you replace phenylalanine 85 or 197 with a spin label marked as R1 here, you reduce self-association. If you replace both, you essentially kill it. Now, if you replace these phenylalanines with alanines, basically removing the phenyl ring, you also kill self-association, making constitutively monomeric arrestin protein. Now, the central graph shows bovine protein. The right-hand graph shows mouse protein. And if you replace the same 2 phenylalanines in the mouse arrestin 1, you also get monomeric arrestin that cannot self-associate. So the creation of this mutant gave us a tool to ask, why the heck do visual arrestins of different mammalian species self-associate? It's not obvious because only monomer binds rhodopsin. Now, before I tell you the data, I need for non-visual people to show you this. So rod photoreceptors have a huge signaling compartment called the outer segment, which contains rhodopsin and all the proteins of signaling cascade. And the rest of the cell is more or less normal neuron with a synapse. Now, if you do retinal histology and stain for the nucleic acids, you can see this thick layer of nuclei, which is called outer nuclear layer. This is the nuclei of rod photoreceptors. And you can see the unstayed on top, unstayed outer segments that don't have any nucleic acids. And they are pretty long and very well aligned, like soldiers on a parade. So you can measure the thickness of the outer nuclear layer, which tells you how many photoreceptors are alive. And you can measure the length of the outer segments, which will tell you how happy these photoreceptors are. So this was done in a mouse that expresses constitutively monomeric arrestin-1 on arrestin-1 knockout background. And because these mice were made were transgenic, you get lines that express different levels of your transgene. So as you can see, if you express reasonably low level of a transgene, the outer nuclear layer and outer segments are okay. But at high expression, you have the thinning of outer nuclear layer and shortening of the outer segments throughout the retina, from central to peripheral, suggesting that photoreceptors are unhappy and die when you have high level of monomer. So these data suggest that the monomer is cytotoxic, and arrestin-1 self-associates to reduce the concentration of this cytotoxic entity in the cell. Now I will tell you the last sort of politically incorrect things. In the field, it is believed that arrestins will shut down G protein signaling and also serve as signal transducers for GPCRs. So, and pretty often the receptor-bound arrestin is called active, like in the introduction, and free arrestin is called inactive. Now, if you look at everything that's published, receptor-bound arrestins activate or facilitate activation of 4 different kinases: SRC, ERK1, 2, and focal adhesion kinase. Now, the free arrestins without receptor input do a lot more. Arrestin-1 binds enolase and inhibits its activity. All arrestins bind E3 ubiquitin ligases, parkin, and MDM. And in case of arrestin-3, it actually enhances parkin-dependent mitophagy. All arrestins bind microtubules and calmodulin. Arrestin-3 facilitates activation in the basal conformation, facilitates activation of JNK family kinases. Arrestin-1 enhances the ATPase activity of NSF and promotes the disassembly of snare complexes and rods. Non-visual arrestins activate small GDPases, RAW-A and RAC. They bind clathrin and promote disassembly of focal adhesions. Again, arrestin-3 binds maternal embryonic leucine zipper kinase and inhibits its pro-proliferative activity. And it also activates SRC family member FGR kinase. And finally, caspase cleaved arrestin-2, and cleavage happens without the receptors with free arrestin, cooperate with caspase cleaved bead in promoting apoptosis of vertebrate cells. So in some, free arrestins do a lot more things in terms of signaling than receptor-bound arrestins. So from that, I conclude that both conformations, receptor-bound and free, are active, just in different signaling pathways. So I guess I'll stop there and take questions.
Thank you, Seva. Thank you for again bringing up the issue of how many wonderful discoveries were made in visual field that really enhanced our understanding of GPCR signaling. I have many questions, but I will start with a very simple one. Sometimes in our lifetime as a scientist, particularly for youngsters, when you discover something or you observe something, it stays with you forever, and you're not able to answer on that particular issue that you have identified. And so one of the observation, which is perhaps absolutely uninteresting to no one except to me, that I haven't been able to solve. When you isolate arrestin, it has incredibly short emission of tryptophan. It's
Yeah.
like 310 nanometers.
Uh-huh.
When you express it, it has 340. And if you warm up arrestin, native arrestin, the fluorescent go to 340. So it's clearly a different conformation that is isolated and expressed protein. Maybe you can solve, since you are a world-class expert in arrestin, perhaps the most knowledgeable fellow in this field, answer on my questions from my postdoc.
Well, I mean, that clearly says that conformation must be different, or at least the prevalent conformation. I don't think the protein exists in one. It exists in equilibrium, but the prevalent conformation in isolated native arrestin obviously is different from what we get from E. coli. But what it is, because when you crystallize, you see the same thing, but crystal structures have their caveats. So I don't have the answer for this question, except that, well, of course, if fluorescence is different, the conformation must be different. It must be quenched in the native arrestin.
All right. We have two questions in the Zoom. Clint McKenna, would you like to start?
Okay.
Hi, Seva.
Hey.
My question is, does the cytotoxicity of the monomeric arrestin come from some interaction with rhodopsin or with some other GPCRs?
I mean, we can exclude rhodopsin because that cell death I showed is light independent. If you rear your mice in the dark, you get the same thing. As to other GPCRs, well, I cannot tell, but basically, rods have so like 5 or 6 orders of magnitude more rhodopsin than any other GPCR that it may be unlikely, but it doesn't mean that it's impossible. I would say it probably comes from interaction of monomer with something else, and I don't know what else. We saw the phenomenon, we don't know the mechanism.
All right. Daniel Palanker.
Hello, Vsevolod. Thank you very much. My question is, what is the mechanism of decreasing ERP, early receptor potential, and ERG wave about 1 millisecond after onset of light? Even the light is continuous, ERP wave ends. Is that related to phosphorylation of M2 or some of the process you described, or it's something else because it's so fast?
Well, the simple answer is I don't know. And I should tell, I mean, we use ERG, but we are not pros in it. Just like we use structures, but we are not crystallographers or structural people. We just collaborate. So I don't really know. It's a fascinating question. And, well, somebody else should answer it.
Okay, thanks.
So Seba, let me go to second question that I had in mind. All of the GPCR have the same topology with the intracellular domain and extracellular domain. There is another receptor that is very similar to GPCRs, adiponectin receptor, which has a flip structure with with 180-degree flip structure.
Mm-hmm.
So the question, obviously, that you cannot answer, but what is maybe you have some hint on that? Why is this topology so well preserved? Why not nature pick different two orientation, except of that one adiponectin receptor difference?
Well, again, I don't know. I know that evolution works on the principle, if it works, don't fix it. So if at some point it happened to flip and was found to be functionally useful, well, that's how we have it.
Hello, this is Jian Zhang from Dr. Fashi's lab. Thanks for the talk. I have a question. Sorry, I'm not didn't full understand.
Okay.
Yeah, I believe the difference.
Okay, so, well, actually, human and mouse rhodopsin has 6, bovine has 7 sites to phosphorylate, and the answer depends on the experimental model. So we use differentially phosphorylated native bovine rhodopsin, and we didn't find any significant change in arrestin binding depending on the phosphate level. As long as it's been more than three or more, it's good. Now, other people used peptides with phosphates in different places, the C-terminus of the bovine rhodopsin, and they found that phosphate in a certain position, don't remember which, actually inhibits arrestin binding. Because we worked with full protein, I prefer, and because it's our data, I prefer those. But it is possible that depending on phosphorylation pattern, you can get different conformations of arrestin-1. It's probably like this is barcode hypothesis for other GPCRs, and there is some evidence suggesting that it's correct. It sounds very inviting, but we need direct proof, preferably structural, even though I just with mouse structures.
Hi, this is David Solon from Polchevsky Lab. I was wondering if you are aware of the crystal structure of Xenopus arrestin-1 that forms a crystallographic dimer that is very similar to half of your solution tetramer. It's a CC dimer. It's remarkably similar, which kind of confirms your data from many years ago.
Well, yeah, it is similar, but I mean, as far as I know, people solved both Xenopus and salamander arrestin structures, and then they looked at the solution dimers, and those arrestins don't form tetramers, only dimers. And they also found, like with bovine rhodopsin, that the crystal oligomer and the solution oligomer are different. So that tells you that whatever you do for crystallography, what you do, you use unnaturally high concentrations, very unphysiological conditions, usually high salt, the concentration of polyethylene glycol that takes away the water and so forth, that forces a different way of oligomerization than you would find in more physiological solutions.
Dr. Gurevich, is Philip Kaiser in the room. Really nice talk. I thoroughly enjoyed it. Can you comment a little bit on the roles of phosphoinositols in control of arrestin function? This is something that's been binding of these compounds been observed to a variety of different arrestins. Do you see a unifying principle here in terms of the control of the function of arrestins by those compounds?
I don't think it's unifying, but I mean, what is certain that arrestin binding requires negatively charged lipids in the membrane or in detergent micelle if you solubilize rhodopsin. Otherwise, arrestins won't bind. And in contrast, GRK1 doesn't care about membrane composition. Now, there are sites in arrestins where inositol phosphates bind, and some were shown by crystallography and some by functional assays. And as far as I know, all of them bind, although in case of non-visual subtypes, arrestin 2 and 3, inositol 6-phosphate promotes self-association, whereas in case of arrestin 1, it rather inhibits it. Although some of the Chris's data suggest that inositol activates arrestin 1 as it probably does arrestin 2 and 3.
And let's go back to the Zoom. Maxence Nakure.
Yeah, hi. That was a lovely talk. Sorry about that. So I was curious about what you thought about these super complexes, which have been the subject of a few high-profile publications where you can get both arrestin and a G protein bound to a GPCR.
Well, let's put it this way. In this super complex, that was beta-2 adrenergic receptor, but the C-terminus was from V2 vasopressin receptor. So it's not a receptor that exists in nature. Plus, in this super complex, there were two antibodies. And if you carefully look at the structure, those antibodies in salt structure interact with each other. So I cannot say, I don't think it was ever shown with any native GPCR. And what we see in structure, well, it has all the drawbacks of structures.
Thank you.
I have yet another question here about the binding of arrestin to receptors. You mentioned that particularly for arrestin-1, there is negatively charged lipids are critical for efficient binding. So how the heck the detergent solubilized complex or mixture can work if you dilute those lipids with detergent?
Well, that is not my work. This is Francis' work with Martha Sommer. They showed that if to detergent solution of rhodopsin, if you add negatively charged lipids, now your rhodopsin, solubilized rhodopsin, can bind arrestin. If you don't, it doesn't bind. So that's how they concluded that you need negatively charged lipids using micelles. We did this with nanodiscs, and there you can just control whatever lipids go in there.
Yeah, but there are tons of other binding assay with gel filtration, as you mentioned, which is of course in detergent, and also the extra meta 2 assay is also done in detergent. And you don't dope with additional negative lipids. Wouldn't that dramatically change the affinity for receptor?
I would say yes, but in extra meta 2, you use very high concentrations of both proteins. So even lower affinity would work. Because the estimate of affinity of wild-type arrestin for wild-type rhodopsin is something like single-digit nanomolar. But if you use millimolar concentrations, you're like 10 micromolar, which is 3 orders of magnitude lower. Affinity would work just fine.
I see. All right, let's join me to thank our speaker